So, okay, let's get started. Topic 8, cell division 2. So G2, second half of cell division. So we're going to go over, we're going to talk about some things that happen in S phase, G2, some more M, and then spindle assembly and the spindle assembly checkpoint. Okay, so activation of the pre-RC into the pre-RC. So the pre-RC is formed in late G1, and then you have some other proteins recruited. You have to have low levels of CDK, so you're going to form your pre-RC in early G1. And then you're going to form your pre-IC in late G1 when you have CDK active. After the pre-IC is formed, you have DNA polymerase that's recruited and that can transcribe the DNA and create mRNA. And then the complex of proteins at the pre-IC is called the replisome. And then, yeah, this origin of replication will fire and DNA replication will begin. And that's when S phase happens. And not all origins fire at the same time. You can have origins firing at a bunch of different spots at a bunch of different times. Okay. And then you also have inhibition of pre-RC reformation. You have this because you don't want to replicate your DNA 2 times. That's just going to create an accumulation of DNA and increase your chances of damage, which you don't want. And you just don't want 2 rounds of DNA replication to happen in one cycle. And then how is the pre-RC inhibited? You have phosphorylation of ORC by CDC that happens during S phase, and then that inhibits the binding of CDC6. You also have phosphorylation of CDC6, which leads to proteolysis destruction of CDC6. And then you also have phosphorylation of MCM proteins, which export them away from the nucleus. So I think she talks about this too. You typically want all of these things to happen. You typically want all 3 of these mechanisms to happen just to really make sure you don't have that pre-RC forming again. I actually think I have a question. Yeah. So what will happen if ORC is not phosphorylated? Do you think re-replication will occur? Yeah. Why? Yeah, exactly. So if ORC isn't phosphorylated, CDC is probably going to rebind again. And then you just want you just really want to make sure that the pre-RC is inhibited. So you're going to go through all those mechanisms of inhibition. And then you also have cohesion and cohesin. So when chromosomes duplicate, they're held together by a protein ring called cohesin. And it's important for spindle assembly and controls the spindle assembly checkpoint. Basically, it's holding your sister chromatids together so they don't like go away from each other too early. And then cohesion holds the sister chromatids together. So I just said, but if cohesion loading does not happen during S phase, spindle assembly won't happen correctly. I think you have a few questions about this on your problem set. So cohesion loading has to happen during S phase. Yeah. Good. All right, and then G2. So in G2, in G2, you just have, oh God, DNA replication is finished, and you're checking for damaged DNA, you're checking for replication, making sure you have replication went through, there's no DNA damage, your sister chromatids are ready to separate, your cell continues growing a little bit, so checking for those nutrients, and you're also going to have activation of M-cyclin CDK. Even though M-cyclin is present, the binding of M-cyclin to CDK does not mean it's active. You have to have some things happen before that. And then this is what needs to happen. So activating M-cyclin CDK is known as the G2 to M transition. It'll happen right before you're ready to divide. So M-cyclin is transcribed and translated in S phase. And just because it's made does not mean it's active. Remember, she's mentioned this Cyclin is necessary but not sufficient for activation. And then CDK is phosphorylated at two different positions. You have CDK activating kinase, so CAC, that puts a phosphate group at an activating position. And then you have WEE1 that phosphorylates CDK at an inactivating position. So basically CAC pushes CDC forward in the cell cycle and WEE holds it back. And then when the cells sense that they're ready for M phase, that DNA is replicated, they're ready to go, they have enough nutrients, they're all set, you'll have a phosphatase called CDC25 removing WEE1's phosphate group. And then PLK1 phosphorylates CDC25 once it's ready for M phase. And then you have a positive feedback loop, leads to the rise of active M cyclin CDK. Partially activated CDC25 is fully activated by phosphorylation of M cyclin CDK. So active M cyclin CDK goes back into the cycle and kind of phosphorylates proteins that'll make it active. Positive feedback loop. And then active M cyclin CDK phosphorylates and inactivates WEE1. So M cyclin CDK inhibits its inhibitor and activates its activator. Okay. And a positive feedback loop is just any time a protein downstream is regulating its own pathway or something upstream of its own pathway. And then this is just a picture to help go with the words from the last slide. And then here you can see that positive feedback loop I said. Active M cyclin CDK goes back in and like fully activates CDC25. So we can go on and remove that inhibitory phosphate at a higher rate. Good. All right. So think about the following. You have a loss of function in WEE1, have a loss of function in CDC25, and a loss of function in CAC.
Loss of function WEE1 being a gain function at some point because WEE1 is supposed to phosphorylate at the end. So
Yep. Yep. Yeah, that's right. What about CDC25? Mm-hmm.
Oh, well, actually the opposite of that. But CDC25 wouldn't be able to remove the phosphate that's inhibiting CDK cyclin, so it wouldn't be, or it would also be inhibited.
Yes. Nice. And then if CAC is mutated so that it doesn't work,
It's the same with the second.
what does CAC normally do? Right. So would that like push it along the cell cycle? Yeah. Yeah. So like you guys said, if you have a loss of function in WEE1, you have nothing holding back the cell in G2, and you're probably going to move on to M phase before you're completely ready to do that. WEE1 is also named like based on the phenotype. I think you guys talked about this as well. The cell that's too small. And then loss of function in CDC25, you have nothing removing that inhibitory phosphate. So you're being held back in G2, even if you are ready to divide. That means your cell might actually grow too much, or it might actually undergo apoptosis if it arrests without like being able to move on. And then if you have a loss of function in CAC, you can't be phosphorylated at the activating position. So you're going to be held back too much. So you won't continue on to M phase. Yeah.
It's all in G2,
Yes.
like that.
Sorry?
Like that, like that.
Yeah. Yeah. It's like at the transition phase. Think of them. Yeah. I like G2 to M transition. Yeah. Okay. And then something in M phase is M cyclin CDK will phosphorylate proteins in M phase that are necessary for cell division. So these are a few of the proteins that Dr. Francis mentions, which are the lamins that are used to break down the nuclear envelope. condensins that condense the chromosomes. And then you have maps and motors that are required for spindle assembly and separation of sister chromatids. So this is something Laura also talks about, that CDK acts in sequential phosphorylation. CDK is usually the first thing to phosphorylate a protein. This is called a process of priming. So it's a priming phosphorylation before a kinase will come and fully phosphorylate it later on. This is an example of CDK acting as the master cell cycle regulator. It also shows why CDK is a great target if you want to halt the cell cycle. Because if you degrade a bunch of CDK or all the CDK, you don't have anything with that priming phosphorylation. So that protein is probably never going to be fully activated. Okay, and then we have some subsections of M phase. So like PMAT stuff. So prophase, you'll have a few things going on. You'll have spindle assembly will start. Your centrosomes start separating. A lot of microtubule networks in the cell will undergo dynamic instability. So they'll be broken down into their tubulin components and they'll reassemble into the spindle. Then you also have your chromosomes will start condensing and your nuclear envelope will start breaking down. You also have prometaphase, which is when, your nuclear envelope is completely broken down. Your chromosomes are already condensed. You have these kinetochores onto the centromeres, and your microtubules are extending from their microtubule organizing center and connecting to each of those kinetochores on the chromosomes. In metaphase, the chromosomes align at the center of the cell. Think of it as like metaphase middle. You also have motor proteins involved to make sure that the chromatids are aligned where they're supposed to be, and your sister chromatids have to have proper attachments to those microtubules. And then once the microtubule is bound to those kinetochores, you'll have stabilization of that microtubule. Okay. Continuing, you'll have anaphase, which is basically the sister chromatids being pulled apart and the spindle pole being pushed apart. So an anaphase is divided into anaphase A and B. A is when the chromosomes are moving towards the pole and the microtubules are shortening. And then anaphase B, you have the spindle pole physically being pushed apart. Then telophase and cytokinesis is the reformation of the nuclear envelope, the cleavage furrow, and then the spindle is basically broken down. So it's the physical division.
I don't understand like really the difference between the two, because like I thought that like when like the chromosomes are moving like towards the pole, like the poles are were like.
It's a good point. So think of chromosome anaphase A as the moving of the chromatids themselves, and anaphase B as moving of the poles, if that makes sense. It also, yes.
So are they happening at the same time?
They can happen at the same time. It just depends on the organism. Some organisms will go through at the same time. Some will go like A and then B. Some will go B, then A. And I think there are some problem set questions that we can go over that'll like help you distinguish between it more clearly. I know that was hard for me to go over that. There they are. I forgot I put them on there. Okay, so anaphase A or B. You have egg 5 sitting on the interpolar microtubules. I realized that, let's actually do this question in 1 second, because I'll have more on like the motor proteins in a second. Wait. I hate that. Okay, let's talk about M phase a little bit. So some vocabulary. You have aster microtubules, which are on the cell cortex. And you have interpolar, so the ones that overlap. And then you have kinetochore microtubules, which are the ones that extend from the spindle pole and attach to each of the sister chromatids. Okay. So your spindle pole, your aster microtubules, your kinetochore microtubules, and your interpolar. That'll overlap with each other. So, and then you have the push-pull theory that is basically stating that the spindle assembly requires lots of opposing forces on each other. So dynein is a -N directed mortar that binds to aster microtubules, so the cell cortex. And it holds onto the cell cortex and holds onto the microtubule and walks to the minus end. This pulls the spindle poles apart. So it pulls these apart from each other. And then egg 5 binds to the interpolar microtubules and walks towards the plus end, which also pulls the spindle poles further apart from each other. So egg 5 sits on the interpolar.
Yeah, I don't understand you there.
The walking?
But I don't know. What did you do? Like pulling it like in towards each other, like, or am I just like?
Hold on.
stupid?
Dynein sits on the minus end and pulls the spindle poles back. Egg 5.
Then it's like closer to like the center zone and like.
So they're kind of like walking in different directions and like the natural forces, right?
Yes, like there are lots of opposing forces. So the dynein sitting on these aster microtubules are providing an outward force. And the egg 5 are binding to the interpolar microtubules. I know exactly why you're confused, but I don't have like the best explanation right now in my head. Walks towards the plus end, pushing the spindle poles.
Cause like, I feel like in my
Oh.
head, like, okay,
Sorry. No, but they'll sit here. It's just exactly what you said. They're sitting here and they're pulling on opposite like directions and they're pulling, that's making these pull apart. Like they're overlapping microtubules, so that's why it provides an outward force.
so like the one on like one grabs the other and like uses like the feet on the other one to push.
Yes.
Okay,
Yes.
so like it's just like holding on to like the microtubule it's on,
Yes.
okay. Okay,
And then it
Yeah.
pulls this way.
that makes,
We'll find some videos.
yeah.
That usually helps.
Okay.
Okay. And then dynein can also sit on interpolar microtubules. Right here. And then like it holds on to one microtubule and it walks on the other and it pulls the spindle poles closer together. So it pulls the ends toward itself. So it's like holding this one and pulling it. All right, now we'll go back to. Okay. Anaphase A or B. Egg 5 sits on interpolar microtubules and basically pushing the spindle pole apart. Is that anaphase A or B?
Okay.
It is B. That one is B. How about the kinetochore microtubule shortening? Yeah, because if the microtubule is shortening, the actual chromatids are being pulled, right? Yeah. For anaphase A or B, it's always good to just see like what's going on. Are the spindle poles moving or are the chromatids moving? Cool. And then we went over this. We'll watch some videos in a little while. And then I think spindle assembly checkpoint. Okay, so cells can sense opposing forces, like pulling and pushing forces on the kinetochores. And once we have proper microtubule attachment, the motors are sitting on kinetochores and walking in opposite directions, provides enough tension. But cohesion will keep the sister chromatids together until the cell senses enough of this pulling force. Once the cells sense enough tension, MAD and BUB will inactivate. The activation of MAD and BUB will release CDC20. CDC20 will then bind APC, which is a ubiquinator, so it tags things for destruction. CDC bound to APC will degrade securin. Securin was holding back separase. So now separase is free to do its thing. Separase will go and cleave cohesion between the sister chromatids. This means that the sister chromatids can be pulled apart. That makes sense? It's good? Okay. APC CDC20 complex will also target M-cyclin for ubiquitination. You need this because you need your nuclear envelope to reform, and you need your spindle to unassemble and your chromosomes to decondense. Basically, this is what allows for cytokinesis and the final parts of cell division. All this makes sense? What if you have a loss of function in CDC20 and a loss of function in securin? Loss of function in CDC20 and a loss of function, I think, in securin. Yeah.
CDC. Which means they'll never be able to degrade security. Which
Yeah.
means, separates from range.
Yeah. So your sister chromatids are being pulled together. So you're arresting at the spindle assembly checkpoint. What about securin?
Or wait, no. Securin loss of function would cause separase. Wait, no, it's okay.
Okay. So securin is holding back separase. Securin is basically preventing the sister chromatids from being pulled apart.
The separase would always.
Exactly. Yes. So you would have premature division. Yes. So first one, if you don't have CDC20, just like you said, you have nothing to destroy securin. You have separase always being held back. So you have an overactive securin basically, and you're arresting at the spindle assembly checkpoint. If you have a loss of function in securin, you're never holding back separase. So separase is always free to go do its thing, cleave cohesin, and the sister chromatids are being pulled apart probably prematurely before they're ready or before there's proper like connect. Oh my God, my like braining, ordering. Yes, you have improper spindle attachments, and then that could lead to like defects in cell division. Yeah. And then that was it. That was all of topic 8. But I'll be here until whenever you guys want. We could go over problem set stuff and any other questions you have about the topic. I'll be here.